Alright, so just a reminder, Mendel published his paper on heredity in 1866, and the scientific community really did little with his work at that time. Mendel then died in 1884, and he had no recognition for his contributions to genetics. But since then, we have rediscovered his work, and it has elucidated or given us more information on things like complete dominance that Mendel studied. And there are some exceptions we've now learned to Mendel's principles. There's things like incomplete dominance or codominance, or polygenic traits, where many traits are controlled by more than one gene. So, since Mendel's time, we found that some alleles are neither dominant nor recessive. The idea of complete dominance, which is what Mendel studied, meant that the dominant trait, if present, always shown over the recessive trait. So, in a homozygous dominant individual or a heterozygote, we saw as the phenotype the dominant trait. But we now know that's not always the case. All traits are not inherited by complete dominance. So, some more complex inheritance patterns that we're going to look at are incomplete dominance, codominance, multiple alleles, and polygenic traits. So let's first talk about incomplete dominance. Now, this is a situation in which neither allele is dominant. They work together, they produce a blending type of phenotype. This is an inheritance pattern in which all heterozygotes have a phenotype that's in between the two other alleles. And that's what produces the blend. The heterozygote with the two different alleles present in their genotype presents a new phenotype where we see a combination of the two alleles, not both present at the same time, but instead blended well together. So it's always going to result if you have the heterozygote in a phenotype that is intermediate between the two homozygotes. And we'll look at some examples. So the alleles with incomplete dominance are only going to be represented by capital letters only. So here are the Japanese 4 o'clock flowers. There are red flowered plants, white flowered plants, and pink flowered plants. The pink flower genotype is a heterozygote because it contains one allele for red flowers and one allele for white flowers. So it's not the same allele, making it the heterozygote. The heterozygote, when we have a trait that is inherited with incomplete dominance, is a blending. Hence, the heterozygote, or the two different alleles here, produce a pink flower plant, which is intermediate or between red and white, or represents a blending of the red and white colors. So, what happens in a Punnett square when we cross the red flower with a white flower? Well, we actually get, using all capital letters, offspring that are all heterozygous and have pink flowers. Because in the situation where we have incomplete dominance, we also have incomplete assortment, thanks to the law of independent assortment. So it's now at play, and this incomplete assortment produces a blending. We can also look at codominance. This is when two alleles both appear in the phenotype. And an inheritance pattern like this is one in which the heterozygote expresses both phenotypes. It's showing both alleles. One of these traits is not dominant over the other, they're both dominant. A good example is the AB blood type. Someone who has blood type AB expresses both the A and B phenotype because they inherited both, and that is a codominant type of inheritance. Whereas if only one was dominant and the other was recessive, or vice versa, you would be just A or just B, or even if it was incomplete dominance and you inherited one A and one B allele for blood type, then you'd have to see some type of blending or new phenotype be produced. But that's not what we see with codominance. So this generally results in a heterozygote that expresses both alleles. Usually we're going to signify these with superscripts. They could be signified with subscripts or small letters below or above, depending on your textbook. A great example of codominance is coat color, hair color in animals, things like cattle and bunnies. For example, we can look at roan coat color in cattle. This is represented by a mixture of both red and white hairs. And if you were to look really close at these heterozygote individuals, even though they appear to have kind of this blend of coat color, when you get really close to them, you can see that their coat color is actually made up of individual white and individual red hairs. Sometimes we have splotches, which are much easier to see, that both patterns are present, white and red, or we have spots, but occasionally you have roan cattle that doesn't have these spots or areas where white and red hairs are present separately, but you would still see the individual hair colors if you got really close again to the specimen. So here's that roan coat cattle inheritance. So codominance, when we're looking at coat color in cattle, if you take a red cow and a white cow and you put them together, they could produce a red and white or roan cow. So this is both alleles inherited together, both possibilities. We don't see a blending. Instead, both coat colors show up on a roan cow. And then there's multiple allele inheritance. We used, for example, blood type as a good example of codominance. But if we were to look at the complete blood types instead of just AB, we would have to look at multiple allele inheritance. Because there's more than just A and B, you could also have the O blood type or AB. So the human blood types are A, B, O, and AB. And that fits under multiple allele inheritance because that's when two or more alleles contribute to the phenotype. So A and B we've talked about are codominant to each other. If you inherit both, you show both in your phenotype. But both A and B are dominant over O. So if you inherit one allele for A and the other allele for O, then your blood type is A. And the same goes for allele B. Now, an antigen is a substance that, when introduced into the blood, triggers the production of an antibody, or a specialized protein in your immune system used to attack and destroy specific antigens. Now, in general, we wouldn't talk about things that are present on our own cells as antigens, because it's usually anything foreign that could cause disease, hence an antibody wanting to attack it. But because for our ABO or AB blood types, humans pre-make antibodies in order to fight off any foreign blood, we consider these surface proteins on red blood cells antigens because they could start an immune response, especially if you're given a blood transfusion. So let's look at the glycoproteins or the surface proteins on the different types of blood cells. So if you're type A and we looked at your red blood cells, you would have the A antigen, or this green surface protein. It's just given as an example, but you would have the A antigen on your red blood cells embedded in their plasma membranes. The allele you would have inherited could be 2A or just 1A and 1O. They would both give you the type A blood type. Maybe you inherited type B. The allele I superscript B is one way to write the B allele. And that would mean that your cells are coated with the B antigen. That is the surface marker that's on your red blood cells. Now, of course, you could have inherited 2 B alleles or 1B and 1O and still had the type B blood type. If you were type AB, you inherited type A and type B surface proteins. They're both embedded in the cell membrane of your red blood cells. And your genotype is always going to be A and a B allele. Or it would be I superscript A, capital I superscript B. That's just another way to write type AB blood type. And if you have type O blood, you have absolutely no antigens related to the A or B glycoproteins embedded in your blood cells. This would be the blood type known as the universal donor, because essentially everything's absent on these blood cells. Because everything's absent on these blood cells, it can be given as long as they're given as packed red blood cells to any of the other blood types without a reaction. So here are the 6 different genotypes that you could have. Again, type A could have 2 different genotypes. You could have 2 alleles for A or just one, and the lowercase I represents the O allele. So two lowercase i's represents the type O blood type, or that particular phenotype. You could have two B alleles, or just one and another O allele, and still be type B. But for type AB, there's only one genotype. So again, 4 different phenotypes. You're either A, B, AB, or O, or blood type. We know alleles A and B are dominant to allele O. So you could have 2A or 1A. As long as the second allele is O, your phenotype will still be blood type A. You could have 2 B alleles or 1 as long as the second allele is O, and your phenotype will still be blood type B. The only way you can have blood type AB is to inherit an A and B allele from your parents, and the only way that you can have blood type O is to inherit 2 recessive O alleles as your genotype. Here's a summary table. You can see it gives you the blood type, the possible genotypes. It shows you a pictograph of the different glycoproteins or surface markers that would be on the individual blood cells. So for type A, you have type A surface markers. Type B, you have type B surface markers. Type AB, you have A and B surface markers. And for type O, you have none. Now remember we mentioned that for the ABO blood groups, antibodies, or things that will attack antigens, or unknown surface proteins, are produced pre-made in the human body. That doesn't happen for very many surface markers at all that we see in the human body or as a part of an immune system reaction. So what you generally have in your blood plasma, which is the fluid the blood cells are in, are the antibodies that are opposite your surface proteins. So if you have type A blood, you have type B antibodies. If you have type B blood, you have type A antibodies. So you're going to have antibodies against the blood type you don't have. Because if you have type A blood, you don't want to be given type B. So your body has antibodies to attack the type B blood. For type AB, because you have A and B surface markers, you have no antibodies against these surface markers in your blood plasma. If you're type AB and you had A or B antibodies, they would attack your own cells. Someone with type O blood would have both A and B antibodies because they don't have either A or B. So if they received A or B, their body thinks that's foreign or unnatural or not supposed to be in their body. So again, even though type O is the universal donor, you generally would only donate packed red blood cells without the plasma because the plasma of a type O individual would actually contain quite a lot of antibodies that could attack or cause a negative transfusion reaction when blood is given to a recipient from a donor. The type AB person would be the universal recipient. Because they have no antibodies, we don't have to worry about separating their plasma or taking that into effect or account because of them being a recipient for a blood transfusion. They'd have no antibodies to attack anything, so they could receive any blood type. So if we're looking at how common are the different blood types, type O is actually the most common. And it's more common to be positive than negative. And there's another class that we could really get into and look at why is someone positive versus negative. It has to do with the presence or absence of the D antigen. If it's there, you're positive. If it's not there, then you're negative. But in general, just looking at collectively type O, positive and negative, it is the most common, with type AB being the rarest. So let's talk about a sample problem. If we have a man with type AB blood and he marries a woman with type B blood and her father has type O blood, what are the chances that they have a child with type A blood or even type AB? I'm going to give you guys a moment to think that through. All right, so we know that the man has type AB blood. We know exactly what his genotype is. It is type AB, or you could just write I superscript A or I superscript B. But his blood type is AB. We knew exactly what he has, one allele A, one allele B. And we have a woman with type B blood, which could pose a problem. She could be one B allele and one O allele, or she could have two B alleles. But we're also given information about her father having type O blood. So, what are the chances that they have a child with type A blood or type AB? We know exactly the cross to perform. We'd have the man with type A and B. You put that at the top or the side of your Punnett square. And the woman would have to be type B and O because her father only had type O to donate to her. So, regardless of her mother's blood type, which must have been B something, she is B and O. Those are the two alleles she has. So we'll put those two either the top or bottom. The opposite place you put the man's genotype for your Punnett square. All right, so looking at that, make sure that you have everything set up correctly and then perform your Punnett square. So draw a box. We'll put AB at the top and we'll put BO on the side. And we have 4 open boxes to perform our Punnett square. You bring all the letters up top down, all the letters on the side across, and then you look at the results. So looking at the results, it appears that for them to have a child with type A blood, it's only going to be 25%. Only 1 out of the 4 boxes would result in a child that has type A blood. For type AB, it's also going to be only 25%. The AB, you must inherit both alleles, and it's only 1 of 4 of our Punnett square boxes that have type AB. But it would be 50% probability that they have a child who is type B, whether it be homozygous dominant or 2B alleles, or the heterozygote with the dominant B allele and a present O allele. That's recessive. We also want to look at polygenic traits. These are traits controlled by 2 or more genes, and even though it's easy to give eye and skin color as examples when you're learning genetics, these are actually very complicated traits that are inherited by multiple different genes working together to contribute to the final phenotype. The human height, eye, and skin color are all examples of traits controlled by 2 or more genes or inherited via a polygenic mechanism. We also want to briefly just look at the rediscovery of Mendel's work. It was around the turn of the century, which is the early 1900s, that many scientists rediscovered Mendel's work, his ideas about complete dominance or inheritance, or the fact that these factors were being inherited by offspring, that some were recessive or being masked by dominant or more attributable and noticeable traits. Scientists like Garrett, Sutton, and Morgan throughout the early 1900s used his work to further their own research. So, Gregor Mendel really set up the basis for modern genetics. He is the father of modern genetics because of all the work he did. But Thomas Hunt Morgan also added a lot to our ideas about how genes are spliced or inherited or passed down during independent assortment and processes like meiosis. So he did his work roughly in the early 1900s to the mid 1900s, and he was born in Kentucky. He was a professor of biology at Columbia University, and he worked with the model organism called Drosophila melanogaster. So that's a fruit fly. It is a common organism that's studied. You can actually knock them out with carbon dioxide, and it just puts them to sleep for a little bit. So he won the Nobel Prize in Medicine in 1933 because of his research on gene mapping and sequencing. Why did he study the fruit fly? Well, you can work with large numbers of flies easily. They're very tiny. They also produce many offspring very quickly in short amount of time. They have a very short reproductive cycle, a matter of hours. They also had traits that were easy to study, such as shape of the wings or colors of the eyes or stripes on their abdomen. And they also only had 4 pairs of chromosomes. Because of his work on gene mapping and looking at how certain genes were inherited, he was able to provide information on gene linkage. So Thomas Hunt Morgan figured out that genes are linked to each other on chromosomes, especially if they're really close together. Morgan found that many genes are linked together, and those that are linked together tend to be inherited together, and they tend to hang out or exist in really similar loci or locational chromosomes. And it was determined by Morgan that chromosomes, not genes, actually sort independently during meiosis. So it's the chromosomes themselves as material or condensed DNA that are sorting independently or lining up differently during metaphase 1, not the genes themselves, since genes are located on the chromosomes. And that was vital information. He helped put together information that provided evidence for gene maps. And gene mapping became a way to study the possibility of gene linkage and the inheritance of certain traits that, especially, are polygenic and depend on multiple genes inherited a certain way to determine a final phenotype. So this was first developed by Sturtevant in 1911, and he drew a lot of information from Morgan and his idea of gene linkage. And the farther apart two genes are, the more likely they will be separated in meiosis. And we could go into a lot of information on heredity. We're just going to hit a couple more topics. So suppose you have a purple flower pea plant. Well, you know, because the purple is dominant, that its genotype is either FF or F small f. And the reason we know that is we looked at Gregor Mendel's research. We saw that he looked at, among other characteristics, flower color. And in his example, the purple flowered color was dominant to the white flowered color. And so if it's inherited via complete dominance, which it was, like all the traits that Mendel studied, then the capital F, or the homozygous dominant and the heterozygote, capital F, lowercase f, would be the ones that show this dominant trait, these purple flowers. Well, if you wanted to determine which genotype the plant had, 2 capital F's or 1 capital 1 lowercase, you could perform a test cross. If you had no idea, if you hadn't done the complete lineage and been in control of the experiment, a test cross could be performed to figure out the genotype of a dominant phenotyped plant. And that would involve breeding an organism displaying the dominant phenotype of an unknown genotype with a homozygous recessive organism. So you take the unknown dominant phenotype plant showing purple flowers, and you would cross it with a genotypically recessive organism, an organism showing the recessive trait, meaning you know exactly what the organism showing the recessive trait would look like, 2 lowercase letters. So here we see that if we have a flower showing the dominant trait, we don't know if it's homozygous dominant or a heterozygote, we can take a recessive plant for the same trait, so 2 lowercase p's in this case, cross them, and if all of the offspring are purple versus only half purple and the other half white, or you could say if all of the offspring show the dominant trait versus only some of the offspring in a 1-to-1 ratio showing dominant versus recessive, then you'd have your answer. If all of the offspring from the cross show the dominant trait, then your dominant organism was homozygous dominant. If your offspring only show 50% dominant, 50% recessive, then your original organism showing the dominant phenotype with an unknown genotype is actually a heterozygote. So a test cross can provide that information. And then we're briefly just going to talk about pedigrees. So it's a diagram that follows a particular phenotype through several generations. Boxes or squares are males, circles are females, If you color in the box or the circle, it's an affected individual, whether it's male or female, we would just go by the shape. You can generally use a dot in the middle or a half coloration for carrier males, and do the same thing with the circles for carrier females. If you don't know the sex of someone in a particular pedigree, which is generally going to be a phenotype demonstration or diagram through several generations, you would use a diamond shape. If someone's deceased, you draw a line through it, whether diagonal or straight across. A marriage or a relationship that produced offspring is shown by a horizontal line. Offspring is going to be connected to that horizontal line, and offspring that is more than one is going to be shown by horizontal lines that are connected to an initial vertical line coming off of the marriage or mating line. So carriers are individuals in a pedigree who have one copy of an allele for a recessive trait, but do not exhibit the trait. So they're carriers of that recessive trait, especially if a trait is inherited recessively or shown in the homozygous recessive individual. Pedigrees are generally used to determine whether a trait is inherited, show how a trait is passed from one generation to the next, or even determine whether an allele is dominant or recessive. So here are some example pedigrees. The following pedigrees represent experiments involving the eye color of a certain animal. The only possible eye colors for this animal are blue and brown. Brown is the dominant trait. So if brown is the dominant trait, and the other option is blue, that makes blue the recessive trait. It says a black square or circle indicates the brown eye color. So if we color in the shape, that means we're showing the dominant trait in that individual. A white square or circle indicates the blue eye color. So all the not colored in shapes are going to show the recessive trait. We want to know what are the genotypes of individuals 1-4 in the pedigree shown below. It's also figure 7.13 in your book. So let's find them. All right. So we want to look for individual 1-4. So we have two different pedigrees, but they're numbered together, so families that are related. And we want to find generation 1, individual 4, which the shaded in circle. And we want to know what is their genotype. Well, we're not really sure what that person's genotype is immediately, because they're showing the dominant trait. They could be a homozygous dominant, or they could be the heterozygote. It's likely they are the heterozygote because they have offspring that don't show the trait. The only way that would happen is if both father and mother had the recessive trait or were carriers and passed that down, essentially that recessive trait that showed blue eye color, to half their offspring. So the 50-50 suggests that both individual 3 and 4 of generation 1 are heterozygous for the brown genotype. Whereas 1 and 2, we want to make sure we go through 1 and 4, they might be a little bit harder to determine at first, because you think, oh, well, the female here doesn't have the brown-eyed trait, she has blue eyes. Well, then the male here, the father, he must be homozygous dominant, because the only way all of his offspring would likely have that, if he's homozygous dominant, and the only option is for all of their offspring to inherit at least one dominant trait, therefore showing the dominant phenotype, or the brown eye color. Let's look at example 2. So this pedigree below tracks the presence of attached earlobes through a family's generations. And having attached earlobes is an autosomal recessive trait. So those that are not colored in are going to show the recessive trait. And those that are colored in generally would show the dominant trait. We want to see what they want us to find. It says, if individual 3-6, I'm going to go down to generation 3 over to 6, married a man who was homozygous for unattached earlobes, what is most likely to be true regarding their children? All right, so they are showing the dominant trait. attached earlobes is recessive, so they must have the unattached earlobes. And it looks like it really runs through the family. It's highly likely, based on the results of all offspring having unattached earlobes, that these are homozygous dominant individuals, meaning it's likely her offspring with a man who has also unattached earlobes would produce offspring that all have unattached earlobes. They would just continue going throughout the family. So we would have the options: the children would have partially attached earlobes, which maybe all the female children will have unattached earlobes and all the male children will have attached earlobes. We don't expect to see that 50-50 split. It's probably going to be one or the other when we see that in her family it was one or the other as well. And all of their children would have unattached earlobes. They would have the dominant trait. Makes sense because she's shaded in, and if she married a man that would also be shaded in, it makes sense they'd pass that trait to all of their children. Or the kid of all the children would have the recessive trait, which is the attached earlobes. They show the dominant trait, so that's highly unlikely. So the best answer here is the one selected. All of their children would have unattached earlobes. There's also sex-linked genetic traits, and autosomes are essentially body chromosomes. But everyone has a couple of sex chromosomes. They can be XX or XY. So if you inherit XY as your sex chromosomes, then that person is biologically male, whereas if you inherit XX, that person is biologically female, because the sex chromosomes determine the sex of an individual. Sex-linked genetic traits exist because the Y chromosome does not carry as many alleles as does the X chromosome. The result, then, is that some traits for which alleles only exist on the X chromosome and not on the Y chromosome can produce certain inheritance patterns. That means that for certain traits that reside on the X chromosome, males have only one instead of two, so therefore if the trait is on their X chromosome, they'll express that trait, even if it's recessive, because if they show it on their X chromosome, they only have one. And then, whereas females, they have an extra X that acts as a buffer and may prevent them from showing that trait, especially if it's recessive, but they would be carriers. So let's look here at some X-linked dominant versus recessive pedigrees. All right, so we're going to look down here. So for X-linked dominant traits, we generally see that you're going to have affected daughters, especially if we had an affected father. That would suggest that even with the buffer, it's a dominant trait, so it's showing up on even one of her X chromosomes would make the daughter still have that condition. And then here the sons could have inherited the X-linked disorder, but likely in this scenario got their X from their mother that's unaffected, and therefore they got what they had to get from their father, the Y, and so that's why the sons would be unaffected, because their X that they got would have had to come from the mother, who's not affected, so they'd be unaffected. So an X-linked dominant trait, we're generally going to see all daughters affected, males not affected. X-linked recessive, oftentimes the daughters are unaffected because with recessive, they can use their extra X as a buffer. So we generally have a high incidence of affected sons, but this is a little bit less concrete, and we'd still be making a lot of opinionated conclusions looking at X-linked recessive pedigrees if we weren't aware of the way the trait was inherited. All right, so inheritance of a genetic trait located on a sex chromosome can occur. It can be dominant or recessive. We can look at an example, hemophilia, which is a blood disorder. It is a commonly used example of a sex-linked genetic disorder, especially because it's very well known in those that study pedigrees because of how it went through the royal families of England and Russia. So descendants of Queen Victoria were oftentimes either afflicted by hemophilia or carriers. Because of the way this trait is inherited, the females tended to be carriers, whereas the males that inherited the trait were affected. So you can see that many of the males were actually hemophilic. This was essentially a bleeding disorder which could be fatal, but oftentimes is now treated successfully. And because Queen Victoria married off many of her daughters across the royal houses of Europe and Russia, this disorder passed through their families. It is slightly attributed to the reason that the royal family of Russia was essentially martyred and killed and were succeeded during the Russian Revolution. So, Alexandra, who was the daughter of Alice, which was Queen Victoria's daughter, married King Louis, and she was also a carrier because her father didn't carry the trait. When she married Tsar Nicholas II of Russia, even though none of her daughters were affected, her son Alexis was. And it was because of hemophilia, her son's condition, that she went and talked to the medicine man Rasputin, who was known as kind of a crazy, unresponsible beggar, but he said that he had mystical powers, and he actually did try to treat Alexei, and he just kind of gave him random stuff, and occasionally Alexei would say he's feeling better, and it probably was a placebo effect, and he wasn't actually giving him real medicine. But that made the Russian family look even more out of touch with the starving and very hungry peasants that already hated the royal family and their wealth and how much they flaunted it. And this seemed like an extravagance that just went too far, hiring this basically vagabond. And so that led to the beginning of a lot of pressures for the royal family that led in the Russian Revolution and ultimately their death. So even though it goes into history a little bit, we can look at pedigrees and see how this condition was set up or how this scenario came to play out. We can look at the descendants of Queen Victoria and look at carriers versus those that actually had the disorder and how it was passed down. And this is an X-linked disorder. All right, this pedigree shows, I should say, the Duchenne muscular dystrophy or DMD condition, and it's showing it through several generations. This is an X-linked recessive trait. We can see here that the mother in generation 1 has it, and she passes it down primarily to her sons, but she does have one son marry a woman with the disorder, and they produce children that all have the disorder. Because it's an X-linked recessive trait, this woman could buffer it with a healthy X chromosome, but she has the disorder, meaning both her X chromosomes are affected, and that's why we see both her sons and daughter affected in generation 3. So if we were looking at some question answers, we would see that in order for individual 2, so let's go back. In order for the individual in generation 2, number 2, in order to figure out their genotype, we have to look at the inheritance. If the trait is X-linked recessive, and here they're saying they're shading in for those who have the trait, then this individual would have had to inherit an affected X from their mother, but the other from their father is unaffected. So it is likely that they are a heterozygote for this X-linked recessive disorder. So for them to produce an affected son, she'd have to be a carrier. We're also garnering information off of the fact of generation 3, we see as one of her offspring an affected male. All right, so human genetics is a segment of biology that mainly focuses on health problems and diseases that can be passed from parent to child. This really is what a lot of genetics has turned to. We've looked at gene mapping, gene linkage, we've sequenced the genomes of many organisms, and now we're starting to look at human conditions. We're looking at disorders. We're looking at how that inheritance can be prevented, or at least parents can make very knowledgeable decisions about whether or not to have a child based on their genetics. There are 5 ways conditions can be passed through genes: autosomal inheritance, allele mutation, changes in the chromosome structure, and changes in the chromosome number. So, autosomal inheritance is inheritance of a genetic trait not on a sex chromosome. So this is when a genetic disorder is inherited from one of the 22 pairs of autosomes as opposed to the two sex chromosomes. This would make it autosomal inheritance, and generally we see that if the disease is recessive, an individual would have to have both recessive alleles to have the disease. We see a lot of autosomal inheritance do show complete dominance as the method of inheritance, but that is not always the case. So with complete dominance, that would mean that if the autosomal disease is dominant, an individual must have both recessive alleles in order not to have the disease, because the homozygous dominant individual and heterozygote would have the disorder. A good example is lactose intolerance. It's caused by a recessive allele. So if a person is homozygous recessive for this allele, they're missing a key piece of information that would help build a milk digesting enzyme. And by missing that, they can't digest lactose properly, so they would have the disorder. Being homozygous recessive for a recessive allele or something that's autosomally inherited recessively would give you the condition. So again, autosomal dominance versus recessive. To show the disorder, if something is inherited autosomal recessively, all affected organisms or offspring, or however you want to look at it, would have to be homozygous recessive. All right, so here we can look at an inheritance of lactose intolerance through a couple of different pedigrees. We don't quite know what the genotype is, or it would be hard to figure out for any of the blocks or circles that have a question mark. But here you could see how for the two affected parents, almost all their offspring is going to have it. In that case, we could conclude this son will probably also have it because they would only have the recessive alleles to pass down, so this son must also have it. Here we have one unaffected individual, one affected individual. This is autosomal, so it's not sex linked, so we don't really have to look at male versus female. And we could guess, given that most of the children have it, that it'd probably be about 50-50 for those remaining that we don't have the genotype for. We could figure out more information if the unknowns were to get married and have children. We could compare those results. That would give us more information about the unknown children here. Here, in this third example, we can look at the offspring, information we didn't have in example 2, and that would give us information about what the parents are. Here we probably have a 50-50 split. One of these parents is affected, the other is not, and it's likely that the woman is affected. Since the parents are not, it's much rarer or a little less likely that the son will be affected. It is possible, though, because his sister does have the disorder, and it's likely this male also has it because all their offspring are affected as opposed to 50-50 here. So you would just kind of look, and based on information or relationships that produce offspring, give your best guess for a lot of information. A lot of times without genome sequencing, you can't be sure. All right, there's also disorders caused by damaged genes. And this is when an allele randomly undergoes a change during meiosis. We say that it has mutated. And a gene mutation is just a radical change in the DNA of one or more alleles. Usually it's not positive or negative, it's pretty neutral, it doesn't do much. But only mutations that occur in gametes during meiosis could be passed on or inherited. There's point mutations, amino acid changes, frame shift mutations, and they all are going to affect either codons or essentially the type of codon that gets read at the ribosome. It could be a single nucleotide or 1-point change, meaning one base is shifted. It could be a substitute mutation, insertion mutation, deletion mutation, silent mutation. And a silent mutation doesn't really do anything. A neutral mutation. It might change the codon, but that codon still reads for the same amino acid, so no problem. Deletion mutation can lead to conditions like cystic fibrosis. An insertion mutation can lead to the turning on of the BRCA1 gene, which increases the risk for breast cancer. And a substitution mutation is generally going to be multiple or at least one different amino acid that can produce sickle cell anemia. Frameshift mutations affect all the codons after the insertion or deletion, so they could be more widespread or detrimental to a genome. All right, so here we're looking at types of mutations. A point mutation would be, again, if we change one of the bases in the genome sequence. A deletion would be if you get rid of a space or a part of the genome sequence. An insertion would be if you add multiple or even at least just one thing or base to the genome sequence. And a frame shift would be if you were to, because of a deletion, start reading the sequence differently, and the 3 bases you read at a time no longer make sense for the production of certain protein like they did before the deletion that produced a frame shift. There's also disorders caused by damaged chromosomes, and these are changes in the chromosome structure in which the chromosome loses or gains genes during meiosis, and it's deemed a chromosomal mutation. But generally, when a chromosome has too many or too few genes, the body doesn't understand all of the genetic information, and as a result, some disorder occurs. Sometimes you'll have the deletion or destruction of that chromosome and a kind of repair or fixing period that would occur at the end of meiosis. An example of a disorder that could occur because of a disorder caused by damaged chromosomes is Creutzfeldt, which is sometimes called the cat's cry disease because infants tend to produce a cry that sounds like a cat howling, and it's caused by damage to the 5th chromosome during meiosis, and that damage causes the chromosome to lose, so due to a pretty major deletion, some of its genes. And a child produced from this damaged gamete will experience mental and physical challenges, so delayed development, and one physical change is abnormally large larynx, and that's what makes the infant's cry sound like the meow or howling of a cat. Other types of damage: duplication, inversion, translocation. Duplication is extra copies of a gene produced on a chromosome. Inversion is when a piece of a gene is reversed, so it's inserted backwards into the chromosome as opposed to how it's supposed to be. Translocation occurs when 2 non-homologous chromosomes exchange pieces. Translocation is not always going to produce damage unless that translocation was not supposed to occur as a byproduct of synapsis. So here are what those would look like. Duplication, we have 2 of gene B, and that's going to create a longer chromosome than what is generally read. Deletion, we're going to get rid of gene B. That's going to create a shorter chromosome than what is normally read. Inversion, we're going to flip the chromosome so that it's oriented with some of the genes that were on the bottom up top and some of the genes that were on the top down below. And translocation is where we are going to see a shift of genes where pieces are changed. Here we see the lowercase f go into the longer chromosome and the capital F go into the shorter chromosome. And the same thing, we have that reverse for the E's. Disorders due to change in chromosome number can also have an effect called non-disjunction. So in a gamete, if we have a cell that has more or fewer than 23 chromosomes, it's definitely undergone non-disjunction, which is a situation in which abnormal cellular events, meiosis, lead to either none of a particular chromosome or more than what we needed, more than one of a particular chromosome in a gamete. So here we can see that non-disjunction can occur during meiosis 1 or meiosis 2, and we get different results depending on when the non-disjunction occurs. If the non-disjunction occurs during meiosis 2, we create 4 non-viable gametes and they would all self-abort. Whereas if the non-disjunction occurs during meiosis 1, we can generally utilize or could use 2 of the 4 gametes, and they could go through the process of fertilization to produce a zygote or very early fetus. There's different types of non-disjunction. One of the most common that is studied is trisomy 21, which is also known as Down syndrome. And about 1 in 1,000 newborns in North America have Down syndrome. It is associated with some mental delays, some skeletal development that is abnormal. Generally, individuals with Down syndrome would have short stature and very slack or kind of underdeveloped muscles. So an individual with trisomy 21 has 3 of chromosome 21. This is an abnormal chromosome number, so this is an example of this junction. Now, a karyotype is a picture or a representation of all the chromosomes. The actual picture showing the banding and the general location of genes at particular locations called loci on chromosomes. This whole picture would be called a karyogram. There's also plenty of new gene technologies that can use other organisms to perform certain practical tasks, and doing so, using gene technology to perform certain tasks, is called biotechnology. We have plenty of increased knowledge of microorganisms, DNA, their genome sequences, and we can use that information at a molecular level to enhance our understanding further or make advances on genomes and sequencing and the information we know. A genome is just a complete set of organisms, genetic information encoded in its DNA. It's all the DNA in an organism. In April 2003, the Human Genome Project was completed, and its task of mapping the entire human genome was extensive. It actually was predicted this would take much longer than it did, but the human genome consists of about 3 billion base pairs of DNA and about 30,000 genes contained on 23 chromosomes. That means there's about 6 billion base pairs contained in the nucleus of each cell. 6 billion base pairs. And most of the protein coding DNA in humans is pretty similar to other short tandem repeats or repetitive sequences from person to person. So if we were to compare different individuals, both human, we would find that a lot of the repeating sequences that are all next to each other are similar. And that's what we would use for the FBI's CODIS system. It actually uses 13 STRs. Now, they're generally repetitive and remarkably similar, but they're not all identical. And that's why the FBI would use 13 STRs in order to perform forensic analysis. And gene technologies like restriction enzymes, gel electrophoresis, DNA profiling, PCR or polymerase chain reaction, genetic engineering, recombinant DNA, these are all new types of gene technologies that would all be considered biotechnology. Gel electrophoresis and DNA profiling has actually already evolved to include the use of short tandem repeats, and we now call it capillary electrophoresis instead of gel, and it uses a much smaller apparatus and therefore cheaper. Restriction enzymes are pretty much like molecular scissors. They would be looking for a sequence, they would cut that sequence, and by breaking DNA down into little pieces via a very methodical manner, we can basically create small pieces of DNA that we can fingerprint. So here we can see the results of digestion by restriction enzymes. You can create blunt or sticky ends. Sticky ends are staggered, blunt ends are not staggered. And after cutting DNA with restriction enzymes, gel electrophoresis, now actually capillary electrophoresis is more common, is used to separate the DNA fragments for comparison as a part of DNA profiling and fingerprinting. And this is possible because people differ in the number of STR segments. Not necessarily the sequence, but the number of sequences that are repetitive and next to each other. And so they'd have different length for their fragments based on how they were cut with the restriction enzymes. And someone with similar lengths, say, to a victim, that would help if we're trying to identify them, or if you had the killer's DNA at a crime scene, you could take a suspect's DNA and compare it to the killer's DNA and see if you have a match. Larger DNA fragments tend to move more slowly through gels and even capillary electrophoresis than the shorter DNA fragment. The shorter the DNA fragment, the faster it moves. And because DNA is slightly negative in charge and moves from the negative cathode side or the negative side of the gel towards the positive side, we can generate this movement by applying an electric current. So here you can see the setup for gel electrophoresis. You would have a negative charge on the cathode side. You would apply a mixture that contains the cut up DNA. And then you would have a positive charge at the anode on the other side, and that would pull the negatively charged DNA towards it. The smaller pieces would move faster than the longer pieces, so the larger, longer molecules would be up top, where everything started, the shorter molecules at the bottom. You run everything for the same amount of time, so you want to enter it at the same amount of time, and that should produce banding or spacing that is comparable. So here we are looking at DNA profiling. So we could compare the banding to figure out if a crime scene sample matches a victim or suspect or vice versa. PCR is what is performed if you don't have a lot of DNA in a sample. It copies and amplifies DNA many times over. Very useful for crime scenes if you only have a little bit of the victim or suspect's blood. So this is a process that is now very easy to do in a lab, and essentially PCR mimics the process of DNA replication, and it's just amplified. You would apply heat, you would separate the double helix, you would then add DNA nucleotides, we would depend on Chargaff's rules and complementary binding of bases to each other in order to create more DNA. You would do this over and over again until you had enough of a sample to compare. We can also talk about genetic engineering and recombinant DNA. Genetic engineering involves the use of bacterial plasmids and recombinant DNA. Plasmids we talked about briefly when we looked at different organelles. They're small circular sections of extra DNA that confer one or more traits to a bacterium. They can be reproduced separately from the main bacterial genetic code. Oftentimes they can be given to other bacteria. And recombinant DNA takes DNA from two different species and combines them in one cell. To do so, you'd want to isolate the plasmid or gene of interest, use a restriction enzyme to cut out the plasmid or the gene of interest. You generally would want to create sticky ends so it would stick to something else. And then you'd mix the donor DNA fragment and the cut plasmid together. This would produce recombinant DNA, and you would stick everything back together with DNA ligase. And you put it in a test tube to essentially create more of it. And the finish or the result would be a plasmid with an inserted gene, so that would be your recombinant DNA. So oftentimes plasmids are used to add genes to, and then introduce that small piece of DNA into a bacteria or other organism, so that that gene eventually, the hope is, get combined or added to the DNA sequence. All right, genetically modified organisms. They're pretty interesting. GMOs are organisms that have been genetically altered by humans. We do this all the time. Agribusiness uses GMOs, and fluorescent transgenic fish is an example of scientists using genetically modified organisms to study whether or not they were successful in transferring a gene to these fish. They basically linked the glowing feature to another gene they were trying to transfer to these fish. So if the fish glowed, they knew that they had transferred the other gene that they linked to the glowing. This produces transgenic organisms when you essentially add a gene to their body or their DNA, and transgenic organisms contain the genes from more than one species. We also use recombinant DNA for human growth hormone generation or production, blood clotting factors, hepatitis B vaccine, to generate microbes that degrade oil at oil spills, to neutralize toxic chemicals at dangerous manufacturing sites, and even create nutrient-rich food like golden rice, which has increased amounts of beta-carotene, and that's because golden rice contains a lot of vitamin A, or essentially a derivative that is needed for vitamin A production. And that is it for our PowerPoint on genes and genetics and biotechnology. Please just review the biotechnology information. This PowerPoint is available on Teams. If you're aware of a lot of the definitions, you should be fine. And also go back and look at the different inheritance patterns, including codominant, incomplete dominant, and polygenic,